Recorded.
Okay.
Okay. So thank you, Tom, for the invitation, and hello, everyone. Can I, like, minimize? Yeah, I think it's better this way. I don't know. I don't like those things on my screen, but that's okay. So today we're going to be talking about this kind of new project this idea we came up with to look at SST projections. I mean, six projections in this for the Southern Ocean, and try to come up with something to look at the potential impacts of this climate change on microbial diversity. And okay. So, by the way, this has just been accepted by Frontiers by one reviewer, the first reviewer. So this is good, I think. And we had two goals here. First was to estimate the time of emergence of this anthropogenic signal of SST only across the Southern Ocean. And on the second moment, we tried to, as I said, estimate this potential impacts on microbial communities. For that, we used SST outputs from 18 models. And we used the SHA-1 and Shannon indices from 105 samples collected across the northwestern Antarctic Peninsula. For the methods, we have the time of emergence, which is basically when this force signal escapes the internal variability envelope. And yes, this is kind of operator dependent because it depends on your envelope, how I define it. We used the standard deviation among all for the pi control, the industrial control for all the models. And the machine learning, which is maybe the cool part here, we use this random forest model. It's a what we call supervised model because we provide the classifiers or the forcings and the answers, I mean, the targets. So we had in situ SSTs and values for these indices, the SHA-1 and Shannon, and we trained the machine with that. And then we use the SST output from models to predict these two indices in the future. So, well, this is just so we could get a good look on the kind of SST change we expect in the Southern Ocean. This is the spatial mean for the entire Southern Ocean, and we have these four scenarios. I guess everyone is pretty familiar with those. And it's quite simple. We have this more sustainable scenarios in blue and the other high emission scenarios in orange and red. So yeah, this is those are maps for the time of emergence. I don't know, maybe I should call it TOW, but TOW I think sounds a bit weird. I'm going for TOY. You can correct me on that later, no problem. So here we have TOY maps, right? And I think it's really cool to see when the signal is expected to emerge from the so-called internal variability. And there are a few things that I think are really nice here. If you look at the Weddell Sea and the Ross Seas, everything is seen in white here, except for these really big ice shelves. These are regions where the signal just never leaves the internal variability envelope throughout the 21st century. So I think this is nice. I mean, this ability of some regions to kind of postpone or maybe buffer this warming signal. And in the other end of the spectrum, we have SSP 585 down here, and it's pretty clear what we can expect, right? By mid-century, this green color here. This is mid-century. We have big chunks of the Antarctic coast being hit by this anthropogenic warming. And we have this, I mean, it just we just expect like twice on higher emission scenarios, right? And this tiny rectangle-ish marker here is our actual study area. And here is just that rectangle kind of. those circles represent our sampling stations, the oceanographic stations. And believe me, there are 105 there. And yes, there's this crazy interpolation here. This is in situ SST, but that's not a problem. We're looking at long-term projections and trends. And so I think that's more than okay, right? And this is just a zoom. This is a toy zoom, right? And it makes it clear again that the higher the emissions, the sooner we may expect the signal to emerge, right? And again, we have I think this is quite cool, this not having this anything here in the Weddell Sea, because we know, I mean, this is quite a sensitive area, and it's somehow it's able to buffer this warming at some point, and not forever, of course. And here, this is kind of different. Not sure if this makes too much sense. This was one of the reviewers asked for something like this. He wanted to see some sort of toy anomaly, but toys are dates. So we came up with this idea. So let's look at horizontal anomalies, because it wouldn't make sense to have, like, I don't know, negative times, right? So yeah, actually the cool thing here is that we can compare the mean toys for each scenario. So we're talking about this 20 in the mean, of course, 20 years of difference between the sustainable and scenario and the worst case scenario, right? And now we come to the biological part, and disclaimer here, I'm more into the physical part of the story, but these are maps for the SHA-1 index, right? And to the left, we have the observations. All those 105 observations come together here. We use them to train the model. Like, we use 8% of them to train the model, and then 20% of them to test the model, the random forest model, and then we use that model once it's validated to project these maps using the SST's outputs for each scenario. And well, as we can see here, again, there isn't much change in the sustainable scenarios, and there's a lot of change in the higher impact scenarios. And I think these colors are quite nice. If maybe we could go to Shannon, because here, you know, the green represents like more diversity, and this beige, kind of desert-like beige, represents the loss of diversity. So, and another thing which is really cool here, if we compare these two figures, the observations and SSP 126, they look just like the same. I did compute the amount of change here. It's something around 0.5%. It's pretty close to nothing. So why I think this is really cool, because it's kind of a way to choose the path we should take, right? And again, the other side of the story, in the other end of the spectrum, we have the SSP 585, which kind of raises a big drop in diversity, right?
Can I just ask a question, Marcus?
Sorry?
Can I just ask one question?
Yeah, sure.
So in this figure you have up right now, you're comparing observations with simulations. What's the time period?
Okay, I'm sorry. This is to the right here, we have the projections based on the random forest models to the end of the century. Okay. So it's okay. So yeah, it's just by the end of the century, what we should expect by, yeah, at the end of the century, yeah.
And in the case of the observation, so that's just present day, that's our...
Yes, I'm sorry, yes. And the thing is, we have to combine them together and to look at them like one single group, because we need the more observations possible to make this machine learning training. That's the problem.
So are we comparing the SSP panels to the observations? I wouldn't think so. We're just comparing them. What are we comparing?
Yeah. So here to the left, we have the observations, a map of the observations. Yep. And here, what we are to expect by the end of the century at 2100.
Okay. And so the SST 126, that suggests to me there's been very little change if I compare to the observations. Is that correct?
Is that, yeah, exactly
These
that.
are not anomalies. Okay, I understand. Okay, good.
So, and finally, we came up with this idea too. This is the composition of samples. So we have like 105 samples here. They've been sorted by temperature, right? And in this way, these colors represent the orders. So we have a way to look at the abundance of some groups. And then we train the model with this, with the observed SSTs and the composition. And then we use this model to project for the 21st century using the SSTs from the SSPs. And this is, I think this is quite nice, but maybe this is a bit misleading because maybe the most important message here is that we expect a lot less change within SSP 126 when compared to SSP 585 or 370. And why this may be misleading, because SSTs here depart from negative values. So here for SSP, the mean for that region, right? That's the mean SST for the region. So the thing is, within SSP 126, the mean never reaches, let's say, 0 centigrades. So that's why we won't see these guys here, these alteromonadales appear here. So it's not a matter of the actual abundance we could expect, but more on the amount of change we could expect in each scenario. And I wish I had someone from the biological part here to help me with that, right? And yeah, and I think I'm in time. And just summing this up, and maybe we can talk more later, it's pretty clear that the higher the emissions, the earlier the toys. At this point, we see that these microbial communities do respond to anthropogenic warming. However, it's hard to think about this spatial variability. So at this point, they respond to this warming no matter when the signal is expected to emerge, right? And as we said, SSP 126 is the way to go, right? Because we should expect less changes. And from this last picture, we've been thinking about it, and it suggests that temperature impacts different groups in different ways. And I really wonder, and this is probably the next step, what could expect if we could use more properties like salinity, wind stress, maybe some biogeochemical properties in these machine learned models to predict this abundance or the indices themselves. I don't know. This is the next step, and I think we may find something really interesting. So this is kind of a downside of this third. I'm not, maybe not downside, but we were just looking at temperature, and I'm pretty sure there's more to do with these microbes. And yeah, a bit fast, I guess, and that's it. Thank you.
Thank you very much, Marcos. It's really interesting to see the linking up the physical data with the biological data. One of the, so I'll ask other people to have questions first. So anyone have any questions for Marcos?
Yes, Marcos, may I ask? This is Waliur. Hello?
Yeah, I can hear you.
Yes, this is Waliur from Goa. Yeah, so I have a question on, you know, that you have only considered the aspect of the temperature, SST, but one of the major aspect which could potentially impact, that is the pH, because with the rise of the atmospheric CO2, you see that whatever since the industrial era began, we have the amount of CO2 we have injected in the atmosphere, out of 30% already taken up by the Southern Ocean. So in terms of Southern Ocean CO2 sink, it's huge, and correspondingly, we should expect large change in the pH. So pH is potential, means it's going to be a large change in the Southern Ocean. So did you consider this pH aspect in your diversity, in your calculation or in your model, this aspect? But because pH in the context of Southern Ocean with the rise of atmospheric CO2 is it's a major, one of the major component.
Okay, so no, we did not. We just looked at SSCs, and as I said, I'm pretty sure that with other this is going to be quick and reliable, and that's a good point. pH, we'll be looking at it. Thanks.
Okay, I have a question about the salinity. Is salinity very important for the microbiome? You said you are going to look at that. I don't understand.
Yeah. Yeah, I mean, we are. The thing is, I mean, I'm kind of new to machine learning too, so it's not easy to get these models running. They're completely different from the models we're used to, right, in these numerical models. And yes, we do plan to use, actually, salinity is the next in line. The thing is, we can train them separately. I'm trying to find a way to train these models with all these properties forcing together. That's the tricky part. But yeah, maybe we could look at salinity only and then pH only.
Which one is the most important factor? Temperature, then salinity, or pH? Do we know?
Yeah, well, I wouldn't know. I'm clear here. I wouldn't know. And this is just the first approach. But yeah, I don't know. Maybe someone could help us.
I'm a physical oceanographer, you know, that's why I don't understand it. Okay, thank you.
Thank you.
Any other questions? John?
I do have a couple of questions. These time of emergence calculations rely on a good estimate of the internal variability. So you said you're using, the pre-industrial control simulations. I'm just curious, how variable is the variability from one model to the next? I mean, that's going to be a big determinant in terms of your time of emergence calculations. Have you looked at, model by model?
No, I have not. I have not. We just built this big algorithm, and we just got everything together and came up with this one map of standard deviation that we use.
It'd be a good idea to nice. Yeah, I mean, you might find, as you do in the North Atlantic, that some models have very little variability and other models have, enormous fluctuations. And all models in general, underestimate multi-decadal variability for, physical reasons that we think we understand.
Yeah,
but it's kind of the really feel of these calculations. You really have to be comfortable with the estimate of internal variability.
Yeah, I agree. We have a friend that was looking at sea level rise, and he did something like that, and it was crazy. Models were looking at different ways.
And the other question I had was this buffering you're talking about. I think you said in the Weddell Sea. Do you understand physically what's going on there? Why should that be? And what time of year is this? Are these annual averages or?
Yes, I'm sorry. Yes, annual averages. Yes. And the thing is, the Weddell Sea, it works like I'm pretty sure it's related to the gyre and due to this ice installation and all that circulation. But if you're looking at this trends and at subsurface, and it seems that this warming trends have this delay within the Weddell Sea. Okay. And it's kind of, and it's not, I have talked about this but anyway, it's not completely clear why, but I think that's kind of, I believe that's the gyre kind of isolating that part of
it would be good to, it'd probably be good to nail that down. That's an interesting result. It also suggests that there's a pattern of change. Like you're looking at these calculations are, grid points.
Yes.
What we're dealing with here is a whole pattern of change. So you can do, a pattern-based, because this does suggest that there's, a pattern of change, and you could do a pattern-based time of emergence, which is much more powerful and, something that would be, I'd encourage you to do.
Sure, thank you.
You identified the pattern of change, maybe with an EOF type analysis. Then you get a time series, and then you look at that time series for when it emerges above, 1 or 2 standard or 3 standard deviation.
Yeah, great. Yeah.
Anyways, that's it.
Thanks.
Thank you, John. Yeah, any other questions?
Yeah, one exercise that we did, but not exactly for this machine learning work, was actually look at the time of emergency, but comparing not only multi-models, but ensembles. So another student actually looked at different large ensembles to see if, if we would get the same spatial distribution, and it was right on. It was pretty amazing that the large ensembles. It was 3, right? Marcus was the Canadian CSM and the German one, MPI. And I think the Canadian had something like 100 members. So we did, it was very, very powerful. And compared to multimodal, it was the same spatial pattern. I like the idea to look at pattern based, because I think, this is an important issue to look at the time emergence of signals. But the one thing I was thinking about is in his study, he looks a lot at SST. And so maybe, when you consider the Southern Ocean as a whole, it's something that might, you see the surface manifestation, but maybe, near the Weddell Sea and the white spots, we're talking about subsurface, processes, and, you can actually get that. That would be my explanation for, why, I think. I don't know if you would agree,
Actually,
Mark.
that links into something I was partly, I guess, going to comment on, really, is that there's have you come across a study by Ed Hawkins on the time of emergence of land? I think it was mainly land temperature focused, looking at future projections, and they find that the Antarctic Peninsula region is really one of the last places on Earth where you see a significant signal emerging from the noise. So that's really close to the sort of Weddell Sea. So I think maybe another idea for the future would be to connect together what's going on in, over land and in the ocean, and what common aspects. I mean, there could be impacts from the ocean on the land, and also the other aspects of the atmospheric variability that could be impacting the ocean. Yeah, it's an interesting angle, actually. It's really important.
Yeah, I mean, that's a lot to do. Lots of good ideas. It's going to be a really nice paper, follow-up paper.
Right, I've just seen a message from Irina. What does it say?
Yeah, hello everyone. Can I ask a question? Sorry, I cannot turn on my camera, so I just cannot make a sign. But nice talk, Marcus. Thanks a lot. It's really interesting to see this from connection, the bio and physical connection. I was actually very interested in the observations that you showed quickly. Can you please comment a bit more about this? If you really tried to see how your model is behaving compared to observations, and if you actually have these comprehensive observations, both with the ocean boundary layer changes and if you have actually microbial observations, which I have no idea how to to work with biology, but yeah, I just wonder if it's actually combined observations that you have there. Thanks.
Yeah, yeah. I mean, no, we just have this. All we had was this. I mean, this pack with 105 observations. They're quite close to each other, so we assume they exist under the same systems and processes. Because the problem is, to use these machine learning models, we need lots of data. That's the problem. And in this case, for the random forest, we would need at least 80 samples. And what's pretty close to what we had, because we have to save like 20% of that for training. And so, no, I did not know. Yeah, no validation against any sort of other observations. We just used everything we had to do the training and the testing. And then we forced again this model, this RF model with the CMIP SST. But yeah.
Let me
Maybe,
give my
yeah.
take. I think the idea is like most people, we do physical oceanography. We live in the physical world, right? And, you know, we've been kind of experimenting with the idea of time of emergency. And we can do that quite well. And so, you know, we don't know nothing about the biological world. The simulation is complicated. So, well, could the fact that we can kind of predict or project when the warming signal will come up, how can you transfer this into the biological world? Can we say something about how it's going to change based on some, physical property projection? I think this was sort of like the idea. I mean, how can I use the knowledge that I have with all these, super climate models and try to say something about how the biological world will change? I don't even know if you can apply it to all the, biological properties. Marcus, help me out. If you could have, I don't know, some sort of, phytoplankton distribution, or even, fish recruitment or whatever. But it would be pretty cool if you could just do that. If you can emerge when some sort of fisheries will collapse. I don't know. It's just curiosity.
Yeah. And I think there's another thing that it's really hard to model life, and right. And maybe this would be a good alternative to look at this. Use another kind of model, not numerical models, but like machine learning models. And John, you're right. This signal has emerged in the deeper layers already. We have seen, yeah, but the surface, for some reason, is kind of postponing this.
Okay. Right, it's 25 to 5 now, local time here in the UK. Any last questions before we close that wrap up? Okay, in that case, I'm going to stop the recording now.